Hi everybody, my name is Elaine and I work for the Sault Ste. Marie Innovation Center. I'm also a research associate at Algoma University, which is where we are today. What we're going to do is a short experiment that tests water quality to see where pollution might be coming from. Now before we start any experiment, and before we talk about what the experiment is, there's something that we need to make sure we do. First, we have to clean our station, clear away the clutter, wipe off our counters. Make sure all that's done before you set anything up. And that is what I've done before this recording has started. Second, you'll notice I'm wearing protective equipment. I've got a lab coat on, I've got gloves on my hands, I've got goggles on my face. So this is the standard equipment that you'd want to wear to protect yourself in a lab environment like where I am. However, at home, because these kits are meant to be used at home, you should at least be wearing some kind of protective eyewear, whether it's regular glasses, sunglasses even, or if you have goggles or would like to purchase goggles, they're fairly inexpensive. So at least you should be thinking about some eye protection in case you splash anything in your eyes. We have a river that flows into Big Lake. The people in the town of Lakeville get their drinking water from Big Lake. However, they are concerned that their drinking water is contaminated, making it unsafe to drink. When they look upstream, there are three possible sources of contamination: a dairy farm, a landfill, and a factory. You have been hired to figure out where the pollution is coming from to make their drinking water safe. You start by taking 4 samples of water. Sample A comes from the start of the river, called the headwaters. Sample B is taken from below the dairy farm, sample C below the landfill, and sample D from the lake between the mouth of the river and where the town is located. Now, did you know why you take these samples from those locations? Well, sample A allows us to see what the quality of water is before any unnatural influences. This is called the baseline, which is used for comparing later samples. Sample B allows us to determine how the quality of water has changed as a result of only one source of pollution, the dairy farm. Similarly, samples C and D will let us see if there are any different pollutants that are in the water system that weren't there upstream from the landfill or the factory. Once we know where different pollutants come from, we can then determine how to make the water safe to drink for the residents of Lakeville. So, now we're going to get to our kits. These kits that I'm talking about are pollution kits that you can get from Boreal Science. You can order them online, they ship right to your door. And in them, you will find a sheet that shows you everything that should be in the kit. So you want to first take a look at this, lay everything out in front of you, make sure, number one, you've got everything you need, which you should, and also that you haven't accidentally dropped something on the floor or misplaced it, so that when you're doing your experiment, you're not rummaging around looking for stuff halfway through. So make sure you've got everything. The second thing you want to look at is, in this particular kit, it's the teacher information package, and there are sheets in the back called safety data sheets. And these are important to make sure you review, because they tell you exactly what the materials are in your kit, so if there are any chemicals, which there are, what those chemicals are, what are the potential health risks, and in case of an emergency, how do you deal with it? So if somebody swallows it, or if you spill it, or if you get it in your eyes, what do you do to keep everyone safe? So this is a very important thing to review. Also, of course, we've got our instructions. Make sure you read these in full before you begin your experiment. That way you're not accidentally overlapping steps, or if you have to wait a certain amount of time to do one thing before you do another, you're aware of that and you can plan for it. So review those instructions, make sure you understand them before you begin. And finally, we've got our work booklet. So in this booklet, you'll find a brief description of the experiment that we're about to do. They'll also ask you questions about each material we're going to test. So your nitrates, your PCBs, your E. coli. They'll ask you, where do you think the source could be from? And what do you think the impact of that pollution could be on your community that you're talking about? So in the back of this, you will find a table. And in that table, as you're going through each of these steps, you're going to want to write the results of your measurements. All right? So for each of the pollutants, nitrates, lead, PCBs, and E. coli, for each of your 4 sites that we've taken water samples from, what is the result of each of those tests? And from that, we can then talk about where the sources of pollution are and what the community could theoretically do to try and improve their water quality. So the first test we're going to do is for nitrates. Now we've got our nitrate indicator paper in here. There are 4 slips of paper, and we've got each of our water samples that we're going to be dealing with. So they are labeled. This is sample A, so that's from the highest part of the stream that we've measured. And all we're going to do is we're going to take one little strip out at a time, and we're going to measure each of the water samples. So we will take one little drop of this here like that. I'm moving it a little bit so that I can get water onto the strip. There we go. And I'm going to put my strip on my page just to keep it clean and not to mix it up with anything else, so I know where it's going. I can see it. It's not getting misplaced. Now water sample B. Good. So I have my 4 strips on my page. Now what you will see on your sheet, I don't want to lift it up too high, is you'll see in the instruction box a bar with different colors in it. And I'll show you a close-up of this in a minute. Okay, and what you want to do is you want to try to match those colors that you see on your strips with the color on the bar. That'll tell you whether your sample is safe, sort of safe in a small, as long as you consume it in small amounts, or if it's definitely not safe to consume for drinking water. So let's do a close-up of this, and you can see where the colors lie for each of the samples. Here you see the strips ordered from left to right, samples A, B, C, and D. Sample A at the far left doesn't show any color change, and so it was placed at the start of the spectrum below. The sample is safe. The color shown by sample B tells us that it falls somewhere in the middle of the spectrum and is within the unsafe zone. The colors shown by samples C and D are almost identical and also fall within the unsafe zone. However, the color differences between sample B and the last 2 samples also tell us that sites C and D are more heavily contaminated than site B. Okay, so for the PCBs, we are going to take our water sample, each one, as well as the matching pipette, is what this little dropper is called. This says sample A on it, so I know it goes with my sample A of water. And we are going to put, we would want to double check before we do this, 2 drops, okay, onto our PCB test strip. Now this test strip is chemically active, so once we put the drops on there, it'll have a color change if there's a reaction, if there are PCBs present. So let's do 1 and 2 drops on from sample A. Put the remaining water back in our tube so that we can use it for the next part. That's our sample A. Now we've got sample B. So 1 drop and 2 drop. There we go. Sample B. All right. Sample C. Make sure we've got the right pipette so we don't confuse ourselves later. 1, 2. Okay. And then our last one, again confirm, even though there's none left, we still want to make sure we haven't made mistakes along the way. Water sample D, sample D. 1 and 2. Good. All right. So we've got our droplets on our PCB test strip. We're going to leave them here for about maybe 10-15 seconds, let the water absorb into the strip, and then we'll have a look at what the color is. As you can see here, PCBs were not detected in samples A or B, but they were detected in samples C and D. Now for the last two tests, you're going to want to do this one at a time. But to save time on this recording, I've gone and put 4 drops of each water sample, A, B, C, D, in each of these little wells. And I'm going to now put 2 drops of each of our solutions, the lead and the E. coli solution, into each of these samples. There. So we've got our lead indicators and our E. coli indicators. Now I'm going to put these on our instruction sheet because there is a color gradient on this sheet and it has a white background. So when you want to look at a color of a fluid that you put an indicator solution into, you always want to hold it up against a white paper or some kind of white background so that you can see the truest color that you can possibly get. Alright, so I'm going to put these on here, and I am going to take a photo from above of each of these so that we can take a look at them up close. So when we look at our samples A, B, C, and D for lead, we can see that there hasn't really been a color change in any of the wells, and therefore lead has not been detected in any of the samples. And so when it comes to lead, all of our sample sites are safe. Now when it comes to E. coli, you can see that 3 of the wells have changed color. And so we don't really want to look at the shade of blue. What we want to know is whether or not it's safe or not safe. We're not measuring exact levels of E. coli because we don't have the tools to do that here. So based on whether or not we've seen a change, you can see that sample A is safe when it comes to E. coli, and samples B, C, and D all have E. coli contamination and therefore are unsafe. Now we can put them all together and get a better picture of what our water system looks like. So when it comes to nitrates, our first site is safe, the next 3 are not. When it comes to PCBs, the first 2 sites are safe, the last 2 are not. When it comes to lead, all sites are safe. And when it comes to E. coli, the first site is safe and the next 3 are not. So now that our experiment has been concluded, we can take off our protective equipment. So in the case of gloves, you want to take them off from the inside out, flip them inside out, so that your skin doesn't come into contact with any of the potential chemicals on them. I put all of the materials that we used in the experiment in this little Ziploc bag. This is the baggie that came with the kit. This is where everything came in when I opened it up. And so I seal it to make sure that when it goes in the garbage can, it's not going to contaminate anything or spill and become a problem. Now I can throw this out of my household garbage because this particular kit doesn't really have anything super dangerous that can't be thrown out like this. I wouldn't be pouring anything down your sink unless it says specifically in the instructions it is okay to wash in your sink. Other kits may have other chemicals that you cannot throw out in your household garbage. So make sure you read those safety data sheets, you read the instructions in the kits, and they will tell you if something needs to be disposed of in a very specific way. Now that we have the results of our tests, let's take a look at the sources of each type of contamination. As we saw a few minutes ago, nitrates were found at all sites in gradually higher amounts. PCBs were found at sites C and D, and lead was not found at any site. E. coli was found at sites B, C, and D. Site A did not show any contamination and served as a baseline for comparing what we see downstream. Because the dairy farm is the only potential source of contamination between sites A and B, we know that any differences between the samples is likely coming from there. So if we compare the 2 sites, we can conclude that nitrates and E. coli entered the water as a result of the farm. Similarly, if we compare sites B and C, we can see that even more nitrates are present and that PCBs have also entered the water system. Because the landfill is the only thing between the sites, we can conclude that it added these contaminants. However, since we aren't measuring exact levels of E. coli, we cannot determine whether the landfill added any of this bacteria after the dairy farm. Finally, we can see that there are slightly more nitrates at site D than at site C, so we know the factory added to that contamination. However, we cannot determine if the factory added any further PCBs or E. coli contamination without measuring exact levels. But what about the lead? So community members were concerned about lead in the water because they had sent samples from their taps somewhere to get tested, and it came back that lead was among the things that they had in their water. This is why they hired you, was to try and figure out where this lead was coming from. But we did not find any lead contamination in the river or the lake. So where is this lead coming from? We can conclude that the only source of lead that is coming into this water system is from the pipes in the community members' homes. Now that we know that there is contamination in this lake that we are testing in this fictitious scenario, the question becomes, what do we do about it? Now, we know what the sources of pollution are. We know there's different pollution coming from the dairy farm, from the landfill, and from the factory. So what are the different things that each of those places can do in order to prevent pollution from entering the waters? First, let's talk about dairy farms. If cattle are free range, one of the first things a dairy farm can do is install fencing to ensure that the cows are not able to access the stream directly. This is called cattle exclusion fencing, which can come in a variety of styles to suit different budgets, farmer needs, and terrains. Keeping the cattle out of the waterways prevents them from going to the washroom directly in the water, which reduces E. coli contamination. To help with both E. coli and nitrates or other nutrients, vegetation should be planted along the riverside. The areas along a water body are known as the riparian area or riparian zone, where plants act as natural filters to pull many types of contamination from the ground before it reaches the water. Also, planting riparian zones helps to prevent erosion, which helps the landowner while keeping the waters healthy. Similarly, buffer strips can be planted in strategic areas across the farm to improve the area's ability to remove contaminants before they enter the waterway. Buffers often help to keep the soils healthier as well. The buffer strips in the image on this screen are called contour strips, as they follow the shape of the ground to help capture contaminants where they would naturally go as water runs along and through the ground. Next, let's consider the landfill. Depending on the design of the landfill, it could also make use of vegetation buffers like the farm. A landfill can also build a berm to help prevent waste from blowing into the waterways or getting there through stormwater. The berm on the screen is a combination of blown concrete called shotcrete and an earth berm towards the back. Many landfills also have a system to collect and store contaminants that wash or degrade into the ground, called leachate. Once collected, leachate is either pumped into a storage tank or a holding pond and is eventually transported to a wastewater treatment plant that is equipped to process this type of wastewater. So we've got our dairy farm with its cattle fencing and vegetative planting, whether it's riparian or buffer strips. We have the landfill, which can also plant vegetation as well as put up berms and collect leachate. So what can the factory do? Well, there are a variety of filters that can be put onto different outflow pipes to make sure that whatever contaminants are in that system can be pulled out before the outflow enters the water system. And so we would recommend that the factory puts in a filtering and storage facility for wastewater. Lastly, let's not forget the houses. They should be removing their lead pipes and replacing them with a safer alternative. This brings us to the end of our experiment. If you're interested in these test kits, I'll put a link up on the screen in just a moment where you can go and order one yourself, as well as a link to the website of the Sault Ste. Marie Innovation Center and Algoma University. If you've got any questions about what we talked about today, or you want to know more about water quality, I would be happy to talk more about it with you. My email address is below and will also be on the final screen. Thank you very much for watching, and I hope you enjoyed it.